Morning students, today you are going to study about vegetative propagation in that modern method. Before entering into the new topic, you can recall what I have taught already. Vegetative propagation, the part of the plant can able to grow into a new plant, that is vegetative propagation. There are two methods, natural method as well as artificial method. In natural method, I have already taught vegetative propagation in root, stem and leaf. Then in artificial method, there are two types. First one conventional method, second one modern method. In conventional method, there are three types. First one cutting, second one grafting, third one layering. Now we are going to see about modern method. In that, first important point is totipotency. What is totipotency? The genetic ability of a plant cell to produce the entire plant under suitable condition. This is very peculiar for the plant only. What is tissue culture? Growth of plant tissues in special culture medium under suitable controlled condition. We are growing the plant tissue, that is called tissue culture. Then what is micropropagation? The regeneration of whole plant from a single cell, tissue or a small piece of vegetative structure through the method tissue culture, that is called micropropagation. Micropropagation is one of the modern method of vegetative propagation. Let's see advantages. First point, plants with desired character can be produced. Second point, all plants produced are genetically identical, and we can carry out this in any season to produce plant, because some plant we can get particular period, but by using this method we can produce all plant in all season. Then if any rare and endangered plants are there, means we can produce by this method more number, and at last disease free plants also can be produced. First point, advantages, plants with desired character. Then all plants are same, then we can carry it out in any season to produce plant. Then rare and endangered plants can be produced. Then disease free plants also can be produced. Next, what are the disadvantages? Its first point, cost effective. First point, cost effective. Second one, sterile condition is very important, so that also add cost. If any new disease or changing environmental condition mean the whole field, all the plants will be affected. Sometimes callus, a group of undifferentiated cell during tissue culture, undergo genetical change which are undesirable for commercial use. It gives some unwanted character, so that time also it is unuseful. So here disadvantage, cost effective, sterile condition is needed, so it add cost. Then if any new disease come, means it will affect whole field. Then callus undergo genetical change which are undesirable for commercial use. Students, I will give you some questions. You can study thoroughly. We can move into the next topic. Sexual reproduction, it involves production and fusion of male and female gametes. The production of male and female gamete is gametogenesis. Fusion of male and female gamete is called fertilization. The reproductive part of the plant is flower. Yes, the condensed shoot only modified for reproduction is nothing other than flower. Let's see parts of the flower. Outer hole is calyx, it is made up of sepal. Next layer, corolla, it's made up of petal. The next male part, andrecium, it consists of anther and filament. Female part, it consists of stigma, style, then ovary. Ovary only includes ovule. Here the calyx and corolla, it is just helping for reproduction, so we can call that as a accessory holes. Andrecium and gynoecium only essential for reproduction, so we can call that as a essential hole for formation of male gamete as well as the female gametes. Next we can see the stages of sexual reproduction. Pre-fertilization, fertilization, post-fertilization. First, pre-fertilization. Pre, I mean before. Before fertilization, what is the need? Yes, there is a need of formation of the gametes. First one, male reproductive part, andrecium. Here first we can take Anther primordium. It's nothing other than a group of cells covered by epidermis. Then here some cells will be specialized with the nucleus, that is called archisporial cell. This archisporial cell undergo division and it will result into two cells. This is parietal cell, this is sporogenous cell. This parietal cell undergo repeated division and it will result in the anther wall, that is tapetum, next middle layer, then endothecium. From inner to outer side. From outer to inner side is endothecium, middle layer and tapetum. These are the anther walls and it is covered as usual epidermis. Next, the sporogenous cell undergo repeated division and it will result in the sporogenous tissue. The last generation is sporogenous mother cell that undergo meiotic division and it will result in the pollen tetrads. That pollen tetrads will released as a microspores. Then that microspores at maturity it will become a pollen grains. Sometimes in some plants, for example Calotropis, the pollen grains, especially the microspores, are not liberated freely. They are all joined together and it will give like a structure pollinia. Here I am showing you Calotropis. Here the two lobe-like structure, this is called pollinia. All the microspores are joined together. This structure is called corpusculum. The tube-like structure is called retinaculum. So this is pollinia of Calotropis. Next we will see about the TS of anther. TS of mature anther. Here the mature anther, it looks like a butterfly shaped structure. It consists of bilobe and mature anther is a tetrasporangiate type. It consists of 4 sporangium. Let's see one by one parts. Here this is connective tissue, this is epidermis, then this is endothecium. The next two layer is middle layer, then the last innermost is the tapetum. Then this is the anther cavity inside pollen grains. Then this junction is called stomium. This junction is called stomium. Let's see one by one. First, anther wall. Here it includes epidermis, endothecium, middle layer and tapetum. First one, epidermis. It's a single layer of cells helping for protection. Second one, endothecium. It is also a single layer of elongated cells. Here, inner walls develop some bands. Then cells are hygroscopic in nature. In some plants, that band-like thickenings were absent. And even at the junction of two sporangium also, the thickenings are absent. This is called stomium. This is called stomium. It is helping for dehiscence of anther at maturity. Dehiscence is nothing other than releasing anther. Next one, middle layer. Here 2 or 3 layers, that is middle layer, and it is epimeral. What's the meaning of epimeral? It is short-lived. Last one, tapetum. This is tapetum. The innermost layer of anther wall is tapetum. Here it attained maximum development at tetrad stage of pollen grain. There are two types of tapetum: secretory, invasive. Secretory tapetum, otherwise called parietal or glandular. Here the cells are rigid in nature. It does not lose its shape and it is helping for nourishing the developing microspore. But in invasive tapetum, the cells doesn't rigid, it loses its original shape, and along with the pollen surface, it will form a plasmodial like structure. So we can call this as a periplasmodial type. There are two types of tapetum: secretory tapetum, invasive tapetum. Next, what is the function? First point, it is helping for nourishing the developing microspore. Then it is giving substance for pollen wall formation. First one is sporopollenin. Second one is pollen kit. Second one is pollen kit. Next, it is producing the important protein that is called hexine protein. It is helping for rejection reaction. What is meant by rejection reaction? See, many pollen grains may fall on many plants stigma. But can all possible for growing the pollen tube? No, it is not possible. When the suitable pollen grain fall on the suitable stigma, then only it can able to germinate. It can form a pollen tube. So in that case only the rejection reaction, if unsuitable pollen grain fall means it will react. Then the next one, anther cavity. This is anther cavity. It consists of microspores in young stage and pollen grains at maturity. You know, microspores are haploid in nature. Then the last one, connective tissue. This is connective tissue. Here it is a sterile tissue, surrounds the anther lobe. It have a vascular tissue. Then it contributes to inner tapetum. Here the mature anther consists of connective tissue, epidermis, endothelium, middle layer, tapetum, pollen grains and stomium. So anther wall, anther cavity, then the last one, connective tissue. Next, what's the difference between the microspore and the pollen grains? Difference between microspores and pollen grain. Microspore, it is the immediate result of the meiosis of microspore mother cell. It want to develop further. But pollen grain is a matured microspore. Its outer covering is exine, inner covering is intine, and it consists of a dense cytoplasm and centrally located nucleus. Exine, it is very thick and it is made up of cellulose, sporopollenin and pollen kit. It is not uniform. Here it is sculptured. Here the sculpture pattern is different for different pollen grains. So by seeing the sculpturing type, we can identify the plant. Next one, intine. Here it is thin and uniform, which form the pollen tube through germ pore. That the germ pore is present in the exine. Next one, palynology. The study about pollen grain is called palynology. Then the shape of the pollen grains will differ. It may be globose, it may be ellipsoid, fusiform, lobed, angular, even crescent shaped. Then the size varies from 10 micrometer to 200 micrometer. 10 micrometer is found in the myosotis, then 200 micrometer cucurbitaceae as well as the dictaginaceae. Then sporopollenin, it is the wall material of the exine. Here it is formed from the tapetum and the nature of the substance is carotenoid. It is resistant to physical and biological decomposition and it is helping for withstand in the high temperature as well as strong acid. That's why we can preserve the pollen grains even for 400 years. Which one is helping for preserving the pollen grain? It is nothing other than sporopollenin. Next one, pollen kit. It is also formed from the tapetum, yellow or orange color. The nature of the substance is carotenoid or flavonoid. It is nothing other than an oily layer on the pollen surface. It is helping for attracting the insects and it is helping for protecting the UV radiation. Which one is protecting from the UV radiation? Pollen kit. Which one is attracting the insects? It is the pollen kit. Which one is helping from preventing from the strong acid? It is the sporopollenin. Next one, the commercial useful substance is bee pollen. What is that? Bee pollen. Melitopalynology. It is nothing other than the study of honey pollen. It consists of high protein, carbohydrate and trace amount of minerals and vitamins. It will be act like a dietary supplement in the form of a syrup and tablet. It is using for increasing the performance of athletes and race horses and even it is helping for healing the wounds. Which one? Bee pollen. The study of bee pollen is melitopalynology. Next, development of male gametophyte. What is the first cell of the male gametophyte? It is nothing other than microspore. Then at maturity it will become a pollen grain. Outer layer exine, inner layer intine, then cytoplasm, then nucleus. First the nucleus, it start to divide. Then this is vegetative nucleus, this is generative nucleus. Above the generative nucleus, a wall material will be found and it will form two unequal cells, one with more amount of food and with vegetative nucleus, that is called vegetative cell. Another with less amount of food and generative nucleus, that is called generative cell. This is the two-celled stage. In majority, nearly 60% of the angiospermic plants, the pollination is done in two-celled stage. After the pollen grains fall on the stigma of the plant, it can germinate and it will form two male gametes. The two male gametes reaches the embryo sac. This is one type. Some other time here, as soon as the two cells formed, the generative nucleus, it will divide and it will form two male gametes. It will become a three-celled stage. At this stage also, it will fall on the stigma of the pollen grains. Here also the pollen tube germinate and it will result in the male gametes can able to reach the embryo sac. Dear students, this is the time you can utilize properly. I have given you some work in the vegetative propagation. Study thoroughly. Thank you.